Welcome back, everybody, to lecture number 8. We're going to talk about viral DNA replication. The more the merrier. As you know, just like for RNA viruses, you have to make more genomes. If you don't, you can't make more virus particles, right? So for DNA viruses, the same thing. You have to copy them. And today we're going to be talking about these DNA viruses on the Baltimore scheme, the parvoviruses with single-stranded DNA. We're not going to really talk about hepatitis B virus because we'll talk about it next time. We talk about reverse transcription, but it is one of those viruses. When it's repaired, then this double-stranded DNA has to be duplicated, of course. And then we're going to talk about adenoviruses, herpes, polyoma, and papilloma, the group 7 with double-stranded DNA. And remember, this is something we talked about last time. Viral DNA replication is always delayed after infection. That's why we had transcription first before this, because you need at least one protein, sometimes many, to get viral DNA synthesis going. And today we're going to see that one or more protein being made. So just like for RNA synthesis, there are universal rules for DNA replication. DNA, of course, is synthesized by template-directed incorporation of DNMPs into the 3' hydroxyl of the DNA chain, as shown here. We use the same image for RNA. All the basic principles are the same. You have a template, of course, a primer, and then the bases are added to the 3' end according to the template, of course, and the DNA is always synthesized in a 5' to 3' direction. The template is copied in a 3' to 5' direction, but the DNA is made in a 5' to 3' direction, and it is called semi-conservative replication because we make 2 daughter strands. I don't know why they're daughters. It's always been called daughter strands. So here is a diagram of semi-conservative and conservative replication, in case you don't know it. So here in the middle is our double-stranded DNA, which has to be replicated. And semi-conservative is when you take both strands and copy them both. So both strands are copied. Very straightforward. Conservative is, as you might think, only one strand is copied. And this is not what we're talking about today. Replication begins at very specific sites. On the template, we call these origins or origins of replication. And we're going to talk about how they work today. And DNA synthesis is catalyzed by DNA-dependent DNA polymerase and many other proteins. Actually, there's more than one DNA polymerase, as you will see, and there's a host of accessory proteins. And this last bullet point, this is pretty much brand new. Last year was the first time I said this. I've said for years that DNA synthesis is primer dependent. But just a few years ago, we found out that some DNA synthesis is actually primer independent, doesn't need a primer, kind of like transcription. Here's the experiment that showed primer independent DNA polymerase. Dogma overturned, right? There was a dogma. I used to say every year, DNA replication is primer dependent. And we're going to see that today. In fact, for the polymerases we talk about, actually they are primer dependent. And that's one of the puzzles of biochemistry, is where the primers come from. And there are a number of issues associated with them that we'll address. But here is a wonderful experiment where we're looking at DTMP incorporated picomoles here. So you can measure incorporation of this single base with time. So you set up a reaction with DNA polymerase and a template, and then you ask whether or not you need a primer. So there are 2 different DNA polymerases here in this experiment. One of them is T7 DNA polymerase. T7 is a bacteriophage, and it encodes its own DNA polymerase. We saw, I believe, the structure of that last time. So here it is without a primer, the orange, no synthesis. And you add a primer with red, boom, you get synthesis. Primer-dependent DNA polymerase. And now we have another polymerase here from a bacteriophage called NRS1. And this is a bacteriophage that infects bacteria that live down at the deep sea vents, those smokers at the bottom of the ocean, right? Big piles of smoking rock from hot stuff coming out of the middle of the earth. All kinds of things live around it, and bacteria live around it. It's just amazing. And there are phages that infect these bacteria. And so, those boats go down and sample it, and so they bring back bacteria, they bring back phages, and this one is called NRS1. For reasons I don't remember, they decided to purify the polymerase from this phage, and they found it was primer independent. So here's NRS1 without a primer in blue, plenty of incorporation, and with a primer doesn't matter. Don't need a polymerase. Dogma overturned. So maybe this is an ancient polymerase, and it evolved to utilize primers. This NRS1 polymerase tends to fall off the template frequently in the beginning of polymerization. It's kind of like RNA polymerase that we talked about last time. And maybe having a primer helps to lock the polymerase on. Someone asked, is this an in vitro experiment or in cell culture? This is in vitro. Purified polymerase, triphosphates, and other things you need for the reaction. The mechanisms of DNA synthesis are very similar to what we've talked about for RNA synthesis. In fact, the polymerases look very similar. As I told you last time, they all look like a right hand with thumb and fingers and palm domains. So here's RNA-dependent RNA polymerase at the bottom where the thumb and the fingers domains touch. At the top is a DNA polymerase. And you can see the active site in the yellow beta strands, very much like the RNA polymerase. There's a double-stranded DNA entering. And here the fingers and thumbs are not touching. So similar structure, architecture, and also two-metal mechanism of catalysis, where the triphosphate that is being added by the polymerase to the template depends on two metals, two magnesiums that are coordinated by two aspartate residues at the active site, and that helps to catalyze the nucleophilic attack reactions that eliminates two phosphates and joins the phosphodiester bond. So very similar to what we talked about last time. DNA replication needs at least one viral protein, sometimes many. So always one has to be made. And that's why it's delayed. And it's delayed also because you have to make capsid proteins later. So you delay. Well, actually, the DNA synthesis is delayed because of this requirement. And so capsid protein synthesis is also delayed because you don't make capsids before you have anything to put in them. And what does the host cell provide? Well, simple viruses require more host proteins because they have smaller genomes. So those DNA viruses with small genomes, they encode one protein that they need to, or that's required for DNA replication, and the rest is provided by the host. And then the bigger viruses, the big genomes, can code more proteins, and as you'll see, we'll talk about them. We'll talk about viruses with small genomes that encode only one protein. And there, of course, the problem is, how does one protein get the DNA replication going? And then we'll talk about viruses that encode a lot of proteins. So where does the polymerase come from? I've already told you this. The small DNA viruses don't encode the polymerase. They encode proteins, which we say orchestrate the host, have the host copy their genome. And that's not a simple feat. For one or two molecules of viral DNA to enter the nucleus, you think they're going to have the cell machinery pay attention to it? No, they're going to be ignored. So they have to have a way to attract the cellular machinery. That's what we mean by orchestrate. We're going to talk about how that works today. And these include members of papillomaviridae, polyomaviridae, and parvoviridae. Now, we have talked a little bit about parvoviridae and polyomaviridae, and we'll talk more about them. Papillomaviridae actually used to be classified together with the polyoma, but then they were separated. And these include human papillomaviruses, very, very important medically, which we will talk about more. And then the large DNA viruses encode most of their own replication systems. These include the herpes viruses, the adenoviruses, and the pox viruses. And we'll talk about examples of all of these today. So what proteins are we talking about here? We're talking about DNA polymerases and accessory proteins. So there, in our cells, in our eukaryotic cells, there are a number of DNA polymerases that have different functions, as you'll see. And then there are other accessory proteins that are needed. It's a complicated series of reactions. These include origin binding proteins. Remember, the origin is where DNA synthesis begins. And helicases that unwind DNA. Exonucleases to cut away the primer. And also enzymes that are needed to make the triphosphates. Small genomes don't encode these, but some of the larger viral genomes do, like thymidine kinase is encoded by herpes viruses, ribonucleotide reductase, DUTPase. So the triphosphates are needed, right? And so there are enzymes that make them, and sometimes the viral genome encodes them. So the DNAs have diverse structures that have to be replicated, just like the RNA viruses have a diversity of structures that have to be reproduced. And here they're shown, the ones we're going to talk about today. We have adeno-associated virus, which is a parvovirus, single-stranded DNA virus, and SV40. So that's 4,680 bases. SV40, 5.2 kbp of double-stranded circular DNA. Then we have adenovirus, which is 35,000. So here's what I mean. These two, 4.6 and 5.2, they only encode one protein, essentially, to get DNA synthesis started. And then we jump to human adenovirus, and it encodes its own polymerase and some other proteins too. Herpes simplex virus, double-stranded linear, and then vaccinia virus, 200,000 bases. And the DNA is double-stranded, but the ends are covalently joined. So if you denatured this DNA, if you separated the strands, it would be a single-stranded circle. Now, a key to our discussion today is going to be proteins that interact with these genomes. For example, we have, all these genomes have origins of replication, which are shown here by ORI. So here for SV40, it's right there. For adeno, it's at the ends. Herpes has a couple of ORIs. And for pox, people aren't quite sure where it is, but it begins somewhere, so we don't show it here. The ORI for AAV parvoviruses is at this end here on the left end, as you'll see. And then we have cellular replication proteins, which get recruited to the ORI via these viral origin binding proteins, which are in red. They help to recruit cell replication proteins. And then some viruses encode their own replication proteins, so the bigger genomes, as you see. All right, so there are two broad mechanisms of DNA synthesis: replication fork or strand displacement. All right, so let's start with the replication fork. These are the viruses that reproduce their DNA, replicate their DNA in this fashion. Basically, it is a fork. You have DNA synthesis on both strands, and the fork moves from right to left. It grows bigger and bigger. It starts at an origin and then grows, and we'll see how that works. Papillomas, palleomas, herpes, and retroviral proviruses. The retrovirus provirus, as we'll see next time, the DNA is integrated in the host genome. It's our DNA synthesis that reproduces the provirus. We'll learn more about that next time. So here we use RNA primers. Those are shown in green. So this is a primer-dependent enzyme. The RNA primer is laid down first, complementary to the template, of course, and then the DNA polymerase comes in and synthesizes the DNA, which is shown here in red and orange. They're meant to show you that they're complementary. Newly synthesized DNA is shown in red and orange here. On the other hand, strand displacement is different. Here you have a primer. You begin at one end of the DNA and you displace the top strand here. So as the polymerase is making the product in red, the top strand is displaced. That's why it's called strand displacement. And this also requires a primer, but it is never an RNA primer as it is for the replication fork. It can be a protein primer or a DNA primer, never RNA primed. So strand displacement, distinct mechanism, and distinct primer. And we will talk about examples of this as well today. We'll talk about adenoviruses in which the primer is a protein, and parvoviruses in which the primer is a DNA hairpin, actually. Now, when you reproduce DNA, at least linear DNA, you have a problem which we call the 5' end problem, and that's illustrated here. So here we have linear double-stranded DNA, such as ours in our cells, and if you reproduce each strand with a replication fork, you end up with two copies where you had one before, two double-stranded copies. And each of these has a primer, an RNA primer here at one end, at the 5' end where synthesis began. That's in green. And you have to remove it because you have to make double-stranded DNA. You can't have bits of RNA around. So when you remove it, now you have a gap. And how can you fill it in? You can't. You can't fill it in because to fill it in by the polymerase, you'd need a primer, so you'd have to put an RNA primer on, then you'd still have a gap. So you have an issue, right, on both of them. And so that's the 5' end problem. In our cells, it's dealt with telomerases, which we'll talk about next time. Viruses have unique solutions to this 5' end problem, and we're going to explore that today. It's one of the things we'll look at. Much of what we understand about DNA replication in our cells and in viruses started with SV40 many years ago. Why? Because you could purify the template. 5 kb template, easy to purify. You couldn't purify cellular DNA very easily. And we found there was an origin of replication in it, as shown here. So here's the viral genome, double-stranded circular DNA. You can make tons of virus. You can extract the DNA and put it in a test tube and do replication reactions. And what was learned is that, here we're looking at a linear version just for simplicity. DNA replication begins at the origin. You get a replication fork which goes in both directions. It's bidirectional. And so now you can see here in this bottom image, we have new strands of DNA, the red and the salmon colored, I guess. And there are two replication forks going in either direction, but it all starts at the origin of replication. And above here is one experiment which proved that replication is bidirectional. And this is an experiment where they took SV40 DNA and they cut it with a restriction enzyme, and they knew exactly where the site for the restriction enzyme is, and it cut it in a way that put the origin offset from one end, as you'll see in a moment. So you linearize this DNA, and then you do DNA replication reactions in a test tube. And then you at different times after your reaction begins, you look at the products in the electron microscope. So these are DNAs, these threads here. You can stain them in the way that you can see them. And if you look at the left, that's the early time point. You can see the tiniest bubble there, right? That's the earliest beginning of this replication fork. And then in the second panel, it's a later time point. It's getting bigger and bigger and bigger. And eventually it's almost the whole molecule. And this shows that it's bidirectional because we have an end to compare it with. We can see that both forks are moving. And that's the beauty of this experiment, is that we can cut with a restriction enzyme. The origin is not in the middle of the DNA. It's off to one side, and we can see the ends both growing. So that's the evidence for this bidirectional replication fork. So here's a schematic of this process. Semi-discontinuous DNA synthesis from a bidirectional origin. So you know what that part is, bidirectional origin. There's DNA in both directions, so we have two replication forks. But what is this semi-discontinuous? Well, because of you have to make DNA in a 5' to 3' direction, and the top strand, you can go continuously in one direction. You can make 5' to 3'. You put down an RNA primer in green, and the polymerase just keeps making a product, and the fork grows. This will get denatured, and there's no issue. And on the other strand, you can do the same thing. When this fork is very small, there are these regions upstream here that have to be made in pieces. Now, here we've already expanded the fork, so we know that these have to be made in pieces. But when the fork is small, for example, you put down a primer, and then you make the product. And then the fork gets bigger because of the leading strand on the other strand. And now what are you going to do? You have to put down another primer and fill it in, right? And then when the fork gets bigger, you have to put down. So this is discontinuous replication. And so you have two of those, two discontinuous, and that's the lagging strand. The leading strand is continuous, so that's where we get semi-discontinuous. They could have called it semi-continuous, I guess, but okay, semi-discontinuous from the origin. And then finally, there's no end problem here because it's a circle. It's going to go around, and eventually, let's say this was the end of the circle here. This leading strand is going to continue. You're going to remove the RNA primer. It'll fill that in. All these gaps can be filled in with no problem. There is no end problem because there are no ends, right? And that's how it works out. So that's one solution, which our chromosomes don't have, of course, because ours are not circular. Here's what an origin looks like. This is on the top, the origin region. So the genome was 5,243 bases in length. And so at one point, it's both base 1 and 5243. And that's the origin right there. And we're expanding it down below. But before I go below and tell you about it, this region has an early promoter right next to it. So origins and promoters are often very close to each other. And here's an enhancer for that early promoter. This area on the DNA is nucleosome free. If you remember, I told you SV40 is one of those rare DNA viruses where the DNA in the particle is wrapped around nucleosomes. But this region is free of them, and that makes sense because this is where the DNA synthetic apparatus has to bind. And if we expand this little region here, which is called the core origin, you see it has interesting elements. And in yellow, there are a whole bunch of 1, 2, 3, 4, 5, 6 of what we are calling large T binding sites. There are 2 of them. And T antigen, or large T, I'll mostly call it T antigen, but there's more than one, is that one protein that has to be made to get viral DNA synthesis going for this particular virus. We'll see how that works. And then we have an AT-rich element. So what's with an AT-rich element? Well, as you may know, AT base pairs are easier to melt than GC base pairs. And so this helps to denature this region so that the polymerase and all the other proteins can get in and start making that replication fork. So origins tend to be AT-rich for that reason. And then we have a palindrome, which is a repeat that is part of this. So that's an origin of SV40. And here's what happens. So the SV40 DNA, remember, gets into the nucleus. So the virus gets in, goes all the way through the ER to the nucleus. The DNA gets in the nucleus, and there it's going to be replicated. But before it can do that, it has to be transcribed, right? It goes the early promoter is active. It transcribes an mRNA. For T antigen, the mRNA goes out in the nucleus, T antigen is made, and then T antigen comes back into the nucleus. We had a nice picture of that last time. And the T antigen then binds the viral DNA. And so here is the viral DNA on the top, just part of it. Here's the minimal origin with the palindrome and the AT-rich sequence, the large T binding site. And we have large T proteins. These are these rectangular wedges here. 12 of them bind around the origin, 6 on each side. That undergoes a conformational change or induces a conformational change in the DNA to help denature it. So T antigen is a helicase. It can unwind DNA. So it begins to unwind the origin. And that's an energy-dependent reaction, as you can see here. And then large T recruits a cell protein called RPA. RPA binds T antigen directly, and it binds to the single-stranded DNA, as you can see here, of both strands that has been denatured by the helicase activity of large T. So that's the very early phase of DNA replication. And again, large T is needed. That's that first viral protein that's made. It helps to denature the origin, and it recruits the first of many cellular proteins needed to reproduce this DNA. And so that's the key here, that one protein that gets DNA synthesis going. And, if the DNA, this SV40 DNA just went in the nucleus and sat there, it would never be seen by the host apparatus because it would be one or a few molecules among many, many host DNAs. So the solution is to have a protein that will bind the viral DNA and recruit cellular proteins, which will enhance its chances of being reproduced. And it works because eventually the cell reproduces this viral DNA. So now the subsequent steps I want to go through with you, if anything, to show you how beautiful this is. And again, this was all worked out with SV40 in vitro in a test tube, and then we were able to extend it to eukaryotic cells. So here we have that large T flanked bubble with RPA bound to both strands. Then the first polymerase comes in called polymerase alpha primase, which makes the RNA primers on this single strand, on both strands, of course, and it will also make short DNA fragments. So here we have Pol alpha primase coming in there and start to synthesize the RNA primer, and then the red is the DNA. And then some other cellular proteins come in, RFC and PCNA, which form kind of a sliding clamp on the single strand. And then that recruits a second polymerase called polymerase epsilon, which is going to make long DNA. So now you have your discontinuous replication. The leading strand is continuous, and then you have discontinuous on the other strand. So again, two polymerases, one for making primers and short DNAs, and then one for making longer DNAs. And all this starts with large T antigen, right, coming in to this area. RPA binds, someone asked, what does RPA do? RPA binds large T. It's recruited to the origin by large T. Large T is denaturing the origin. RPA binds the denatured DNA to keep it single-stranded, and then it brings in the primase. See, primase binds RPA as well as large T. So large T is still recruiting the cellular machinery. And then we simply make long DNA. This replication fork gets bigger and bigger. You have your leading and lagging strands. And eventually you have to remove the RNA primer. You have to fill in the gaps and then ligate all the pieces together to make sure that there are no non-covalent bonds there. And this goes around the whole circle. And this is the machine that does it all. Just amazing machine. Here's one replication fork, and there's our hexamer of large T. We have a primase making the green RNA primer, and then the DNA, the new DNA, short DNA there. There's more RNA primer, so that's the lagging strand, and this is the leading strand here. And there we have polymerase epsilon making long DNA together with these other proteins. And this is, I call it a sliding clamp because it's clamped around the DNA and it moves down it. And this happens, of course, really, really quickly. Now, when you get to the end, or even before you get to the end, that's when enzymes called topoisomerases come into play. So here we have our original covalently closed circular template, right? You undergo DNA replication. We're unwinding the duplex, and then we're making new strands, right? As you unwind this and it gets bigger, the rest of the molecule gets overwound, right? Because this is a circle. We're separating the two strands, and as we make new DNA, the rest of the molecule gets twisted. And it eventually, DNA synthesis would come to a halt if we didn't relieve this torsion. And that's the function of topoisomerase, is one of them. Either one or two, there are two of them in the cell, cleaves one of the strands. And it's kind of like an elastic band that you've wound up. You cut it and boom, it all straightens up. And then you have now what we call relaxed supercoils, and the DNA synthesis can continue. And of course, that just one strand is enough to do that, and it's ligated back together. And so this happens multiple times because as you keep denaturing, you twist it up again. Okay, now at the end you have 2 daughter strands, but they're not going anywhere. They're all intertwined, right? They're like 2 circles linked here. So to get them separated, you need topoisomerase II, which cuts both strands now, and that allows the 2 circles to be separated. And of course, they'll be ligated together because you don't want any double-stranded breaks. So two topoisomerases, these are cellular enzymes that are needed in order to separate the 2 strands at the end, and also during synthesis to relax these overwound regions. The SV40 genome is a circular double-stranded DNA. Which statement is correct? It's correct this time. A, T antigen binds and unwinds the ORI. B, replication is bidirectional from a single ORI. C, the 5' end problem is solved. D, has leading and lagging strand synthesis. E, all of the above. Okay, what do we have here? Most of you got all the above, which is correct. All these things are right. All right, so now let's move to another virus with a different solution, the parvoviruses, and we're going to see how DNA priming works. And here we have this unusual single-stranded DNA, which is in the virus particle right there on the upper left. It has a single-stranded genome for most of it, but the ends are folded into what are called terminal repeats. And both ends are terminally repeated, and that works as shown at the bottom. Here's the nucleotide sequence. So we have the genome, and it turns out it is self-complementary. So it can base pair with itself in this way to form a T structure, and that's the terminal repeat. So it's an inverted terminal repeat, and they're present at both ends. That's the repeat part. So this genome encodes a couple of proteins. There's a rep ORF, and then there's the capsid ORF, the single capsid protein that build the particle, and then a single protein that's processed to give different, or I should say, several proteins involved in DNA synthesis. So how does this work? So the DNA, again, goes into the nucleus, and here we show it at the top, and here's the terminal repeat. It's inverted, which means, right, it's going in opposite directions, A, B, C, A prime, D, A, B, A prime, B prime, C prime, A, D. And the prime simply means it's complementary. So A prime is complementary to A, would base pair with A. All right, so it's inverted, and it's formed in such a way that it can make these hairpins, all right, which you see right there. But it's stretched out so you can see how that's made. As this gets in the cell nucleus, the cell recognizes this as damaged DNA. The cell does not appreciate single-stranded DNA, so it repairs it. Polymerase delta will use the 3' end of that left terminal repeat as a primer and simply makes it double-stranded. And so that is DNA primed synthesis, right? Because the primer is this 3' hydroxyl, which I showed you in this slide. And this is a beautiful primer for a DNA polymerase, and it's a DNA primer. So the host cell does that, so you now have a double-stranded DNA. So that's done by the host cell, but you still need a viral protein. And so that's where this viral protein, which is encoded in the left part of the genome, comes into play. It's called Rep78/68, rep meaning replication. So now you know the function of the hairpin here at the left is to be the primer for DNA synthesis. This protein is an origin binding protein. It binds to the origin at the left end, and it nicks the DNA because this intermediate molecule between after step 2, it's all covalently closed. There's no way that a polymerase can do any more with it. So the Rep78/68 origin binding protein binds to the left hairpin. It nicks it. It's an endonuclease. So now you have a 3' end, which the cell can elongate from. And the first elongation, so there's at D, the 3' end. So the polymerase will go from right to left, very short stretch there, elongating and copying the terminal repeat. So now you have a fully double-stranded molecule with 2 terminal repeats, inverted terminal repeats, all right? And then these ends are denatured. They form hairpins. And now we have, as you can see here, displacement synthesis. There's when you denature these hairpins, the bottom strand in red, there's now a 3' hydroxyl, which is open. The polymerase, again, the cell polymerase will start priming there. It will displace the top strand. As you can see here, it's being pushed off. There's the top strand. And now we have this double-stranded molecule, which looks exactly like the one from step 2, where we began this whole thing. And so it goes through the whole process again. And this displaced genomic DNA will go through the same process as well. And you'll end up copying over and over. And again, there's no end problem here because we are priming with this hairpin. And then in this step, we're restoring the hairpin sequence here so we don't lose anything. That little extra copying there before the displacement ensures that the hairpin is restored. So it's continuous replication. There's no discontinuous replication because it's only displacement in one direction. As I said, the ITR is the primer. The viral proteins are needed for clipping and making this 3' end so that we can get more synthesis made. And that rep protein also binds the origin. So there's no replication fork. It's all by strand displacement. So again, in contrast, the SV40, of course, was a replication fork, but this has an end, so we do strand displacement. And again, one viral protein and the rest done by the cell. So let's get a little more complicated. Let's try adenovirus now. So we're going from 4,000 base pairs to 35,000. So remember, these are icosahedral particles with a linear double-stranded DNA genome inside of them. And there's an origin of replication at both ends. There's an inverted terminal repeat, very much like the parvoviruses at both ends. And there's also a small protein linked to the 5' end of each DNA strand called TP, or terminal protein. So origins at both ends. And this is going to be reproduced also by strand displacement, very much like parvoviruses that we just talked about. So let's see how that works. The first step is, what's the primer? And you might guess it's the protein because that's what's on the 5' end of each end of the genome, and it is. And here in these boxes are illustrated the various steps in the protein priming of DNA synthesis. So here's one end of the DNA where we're going to start initiation. And there is Paul is the viral DNA polymerase. It's a virus-encoded polymerase. And then PTP is the pre-terminal protein. It's a little bit longer than the terminal protein, which ends up on the viral DNA. So the polymerase binds to PTP and adds a C to it. And the C base pairs with a G in the viral DNA near the 3' end. The polymerase then adds a couple of bases, CAT, and then it slips back because this GTA is repeated here, so the CAT can slip back, and then the polymerase starts to do continuous synthesis. It leaves behind, of course, the PTP covalently linked to that first C. So the protein C is the primer. And because the slip back allows synthesis from the very end of the genome, so you don't lose any synthesis, there's no RNA primer to remove, the protein stays on, it's processed, and it becomes a little bit shorter, and that's what ends up in capsids. So that's the priming step. Let's move to the main part here. So this polymerase is now going to do displacement synthesis. It's going to displace the top strand, and you can see in this step, it's moving down. We have some red new DNA made. The polymerase is pushing off the top strand. And as the top strand comes off, it is bound by a protein called DNA binding protein, or DBP. And that's going to keep it in a single-stranded form. Eventually, the top strand is all pushed off. It comes down here in step 4. And then you have a new strand, a new daughter strand. You have a duplex made. And this can go through this synthesis over and over because the ends will be primed. The lower strand will be primed just as it has been in step 1. The top strand is down here. It's coated in DBP. And by virtue of the fact that the ends are inverted terminal repeats, they're going to base pair and form a little double-stranded structure, which to the polymerase looks just like the ends of the viral DNA in the very beginning. All right, so that is brilliant because now the polymerase will come in and start to copy it. Even though it's not a full double-stranded genome, it's got the ends that look like an origin. The polymerase will bind. It will synthesize a copy. It will displace the DNA binding protein, and now you have the other daughter strand. So we have semi-discontinuous synthesis. Both strands have been copied. The original blue and light blue, now we have red and salmon. So protein priming, no end problem as a consequence because we start right at the end here, strand displacement. Here's a nice view of this DNA binding protein just to show, give you some insight into how it works. Here's the three-dimensional structure of the DNA binding protein. It's got a body, and then there's this extended part of the protein, kind of like a plow. And these molecules bind to the single strand of DNA as the polymerase is displacing it, and they probably help denature the double-stranded DNA by that plow inserting into the duplex. The next question is, how is DNA replication of parvovirus and adenovirus similar? A, they both require protein-linked primers. B, replication occurs by strand displacement. C, DNA synthesis occurs in the cytoplasm. D, a replication fork occurs in both. E, none of the above. So how are they similar? All right, we seem to be stuck at 25, so let's see. So the answer is B. Replication occurs by strand displacement. That's what's common between parvo and adeno. They don't both require protein link. Parvo was a DNA primer, the hairpin, not in the cytoplasm. In the nucleus, no replication fork. They're both strand displacement. So it's either one or the other, and none of the above is not correct. Herpes simplex virus, we're getting bigger and we encode more replication proteins. Remember, this was an envelope virus with its nucleocapsid inside double-stranded DNA inside the nucleocapsid, and the DNA is a double-stranded linear molecule, very much like adenovirus, except longer, as you can see here. And it has multiple origins of replication and encodes 1, 2, 3, 4, 5, 6, 7 proteins for DNA synthesis. Primases, 3 primases, processivity protein, an origin binding protein, single-stranded DNA binding protein, much like the adenovirus DNA binding protein, and then this one is encoding the DNA polymerase. So this is unusual because it illustrates for us a different kind of replication, which is the rolling circle replication, and that's why I'm showing it to you. Otherwise, if it were the same as adenovirus, I wouldn't show it to you. But there's no protein linked at the ends, so we must have a different solution to the end problem. So the DNA is injected into the nucleus. Remember, the virus, the nucleocapsid docks onto the portal, docks onto the nuclear pore by its one portal. The DNA shoots out of the capsid into the nucleus, and then it's circularized by a DNA ligase of the cell, DNA ligase 4. It joins the ends. Now you have a circle of DNA. And the next step is to reproduce this. How do you reproduce it? Well, if you were SV40, you could do bidirectional replication fork starting from an origin, right? But that's not how it works. It's by a different mechanism entirely, which is rolling circle replication. So we have a covalently closed circle, and it has to be nicked. It's nicked by an endonuclease, which gives you a 3' hydroxyl. You always need a 3' hydroxyl. And that can be used as a primer to start DNA synthesis. So now the polymerase, the viral DNA polymerase, starts to copy the one strand here. The red is the newly synthesized DNA. The other strand is displaced. And you have to imagine that as this red part goes longer, the blue is displaced. And the more the red goes, eventually, its 3' end will come around here and be put off. And then as this top strand is displaced, DNA synthesis can occur on it in a discontinuous fashion. It can only go 5 to 3'. It can't go continuous in the other way. That's the wrong direction. So you have discontinuous DNA synthesis. Eventually, this keeps going, and you make many genome lengths, and they get cut as they're packaged into the particle, as we will see later. But here are 2 genome lengths, and there's no end problem because this is all, you can remove an RNA primer and just fill it in from the previous DNA. You can keep doing that. I suppose the very last one would have an end problem, but you'd make many, many, many copies before you got to that very last one. So the end problem is solved in this case by this rolling circle mechanism where you make very long concatenamers, we call them, many genomes copied together, and the replication occurs on that. So the question is, what did DNA ligase 4 do? The DNA ligase takes the herpes double-stranded linear DNA and makes a circle out of it. So in the virus particle, as you can see in the upper left, the DNA is linear. And when the DNA comes in the cell, it's circularized by DNA ligase 4, and then it does this rolling circle replication. A concatenamer is many genomes all linked together, right? If this keeps going, you think of it as a roll of toilet paper. It keeps unwinding and more come off. And each sheet of toilet paper is a genome. Okay, that's a concatenamer. Next time you go to the toilet, you're going to remember this because toilet paper is a concatenamer of single sheets. I'm sorry if that's offensive, but it's a good way to look at it because you just pull the sheets off. Of course, if you get 2-ply toilet paper, then it's perfect. You have 2 strands, right? All right, our last virus example is pox virus. A couple of unique aspects for this one. And one of them is that these reproduce in the cytoplasm. They are independent of the nuclear bureaucracy. They have nothing to do with the nucleus. The particle gets in and sets up shop in the cytoplasm. And so it's got to encode all the proteins it needs for DNA replication. So here is an experiment where we have infected a cell with a pox virus, and we have stained the infected cell in 2 ways. One with DAPI, which will stain DNA blue. And you can see here this very blue staining structure. That's the nucleus of the cell. But you also see in the cytoplasm, there's some staining going on there, right? In particular, there's some round areas. So what's that? So the next panel, we stain with an antibody to the viral DNA binding protein. And that is in red. And so you can see that is in the cytoplasm, these little foci. And if you merge the 2 colors, then you can see that in the cytoplasm, we have both viral DNA binding protein and DNA. It's probably viral DNA. So those are the cytoplasmic replication factories that the virus establishes. And you can see there's no viral DNA binding protein in the nucleus. Nothing happens in the nucleus. The NIC is done by a cellular endonuclease for herpes virus. So here's how poxvirus DNA replication occurs. This virus encodes at least 15 viral proteins. We're not actually sure of all of them, at least 15, and then the cell has to provide some. So this is a lot. We start with the genome. Remember, the genome is a base paired circle. You can look at it. It's 1 strand, but it's base paired. The ends are joins. And the DNA replication begins at one end by the synthesis of RNA primers and then continuous DNA replication. You end up making a dimer, which then has to resolve into 2 monomers. And how that happens, we're not sure. How the DNA becomes denatured to allow this to occur, we don't know. We just know that in the end, we get 2 monomers made. They somehow separate. There might be some topoisomerase cleavage. We don't know. But I just wanted to show you that, first of all, this is cytoplasmic, and it solves the end problem because there's no end. This is essentially a circle, just like SV40. Now, we've talked a lot about viral origins of replication. That's where replication begins. Talked about the single ori of SV40, for example, at either end of adenovirus, 3 oris for herpes, 1 ori at the left end for the parvoviruses. And these have a few features in common. They're AT-rich segments. They're recognized by viral origin recognition proteins, a way of starting the DNA synthesis there. And we've talked about the mechanisms. There are assembly points for these multi-protein replication machines, and some genomes have 1, others have 3. Our chromosomes have many, right, origins, because if we had only 1 per chromosome, our DNA would never be replicated in time. Here are some closer looks at origins. I've already shown you the SV40 origin with these large T binding sites, the AT-rich regions. They all have AT-rich regions, right? They have areas that can be easily denatured, so you can get DNA synthesis going. So here's the herpes, ori L, one of the 3 oris. It has sequences bound by the herpes origin binding protein. They're repeated just like the SV40. Have an AT-rich region. And then the adenovirus origin, which is at either end of the genome, of course. It's the end of the DNA that is bound by the polymerase and the terminal protein. We have that yellow is where the origin binding protein binds. Then we have AT-rich regions, so it can be easily denatured in this case so that the strand displacement can occur. The other feature that I haven't told you about is that these origins, these viral origins, often have binding sites for transcriptional regulators because they're often promoters in these regions as well. Here, right next to the SV40 origin, there is which is not shown here, but it's a promoter. And these are the SP1 binding sites from that promoter. Here's an enhancer for it. The ori L of herpes is flanked by 2 different promoters. You can see here by the red arrows. So, and it's also the case for adenovirus, which is not shown here. These are binding sites for transcriptional regulators, NF1 and OCT1. So these are concentrated areas of regulation, both at DNA and for transcription. Recognition proteins are, again, the proteins encoded by the virus that bind the origin. This is really a key feature because it gets replication going. And especially for the small genome where you have to recruit the cellular apparatus. And we've talked about large T of polyomaviruses, which binds the origin. The parvovirus Rep6878 binds the origin at the left end. And remember, when this is double-stranded, that same protein nicks it to provide a primer. For adenovirus, the origin binding protein is the P terminal protein, the PTP, which binds the end and recruits the DNA polymerase to add a C to it and start the priming reaction. So that's shown right there. And then for herpes virus, we haven't talked about the origins, but it encodes origin binding proteins, which is the UL9 protein, and that helps to recruit other proteins there and unwinds it to get DNA synthesis going. And so these genomes have features in common. Missing here is the pox virus because the details are simply not known. These origin binding proteins are structurally very similar. They probably evolved from a common ancestor. So we have here a parvovirus origin binding protein, a bovine papillomavirus one, and then the SV40T antigen. You can see just by looking at the alpha carbon traces that the structures are similar. We have this beta sheet, which is architecturally very similar in both, and a series of alpha helices laid around them. And these are very good at binding DNA. Now, the AAV protein, the Rep6878, has an extra feature. It has a cleft here, which is the active site of the endonuclease, which is needed to nick the DNA to provide a primer and so that you can eventually separate the strands, which is what terminal resolution is. So someone is asking if big T the same as LT? Yeah, large T, LT, and that's just because there's also a small t, which I haven't talked about. I'm not going to talk about, but LT, large T, T antigen. Yep. Here's SV40 large T. This has been called the most studied protein on Earth. I mean, people have worked on this, many laboratories for many, many, many years, and have dissected every single amino acid. And nobody does this, or very few people do this, because it's very hard to get funding for this kind of basic research. But this is really what enables us to do a lot of the things we do today. So here's large T, 708 amino acids, and has all kinds of domains in it. Here's the origin binding domain. It's a very specific part of large T that binds the origin. But we also have parts that bind Pol alpha. You can see one here and one here. It's the ATPase part, which is the energy hydrolysis part, the ATP hydrolysis part that's needed for the unwinding. Here's the helicase activity, single-stranded DNA binding. There's a nuclear localization, signal or sequence. And then there's this part labeled RB. That's retinoblastoma protein binding site. And what that means is going to become clear in a moment. This is actually a T for different polyomaviruses, is a species-specific DNA and origin binding protein. And the virus will only reproduce in the right species. So you can have polyomaviruses of monkeys or primates, in which case the virus will only reproduce in primates. There are murine polyomaviruses where the murine virus will not reproduce in primates and vice versa. And much of that restriction, host range, is due to the T antigen. These pre-initiation complexes where the T antigen is binding the origin, they don't form in the wrong species. And as a consequence, you cannot recruit Pol alpha primase to the template, so the DNA never replicates. Large T also binds and sequesters cell cycle regulators and cause cells to get into S phase. So what's that about? Let's talk about that now, and that's going to involve this little piece of large T that binds RB. Most of your cells in your body do not divide, or they do so rarely. Of course, the extreme are your neurons, which don't divide, and then the other extreme are your gastrointestinal epithelia, which turn over every 7 days or so. But in between, most of our cells are not dividing unless you need them to divide. If you are exercising and building up muscle, then your cells have to divide. But if you're sitting studying, you don't need much cell division. And that's a problem for viruses because they do not reproduce well, or many of them do not reproduce well in quiescent cells. Why? Because if a cell is not dividing, it's not making DNA, and there's no DNA machinery, reproduction machinery available for reproducing viral genomes. Makes perfect sense. So when a virus infects a cell, of course, a virus will infect any kind of a cell, whether it's dividing or not. And if it's not dividing, the virus must somehow produce a protein that kicks the cell into dividing. So it has to induce host replication proteins, get the cells dividing. And this is done by viral early gene products. And we've already talked about some of those before, but now we're going to see how they work. Now, to do this, we have to talk about the cell cycle a bit, right? We have a roughly 24-hour cell cycle where we have a G1 period, which is a period of cell growth. The cell is just divided. It's getting bigger now. We have a S period where DNA replication occurs. And we have G2, which happens just before mitosis or cell division. And of course, during mitosis, the cell divides into two, and each cell gets an equal complement of DNA, of course. So everything is leading up to that. Now you have two cells, and then they go through G1 again. There are actually several checkpoints in this cell cycle so that it doesn't go on all the time. And one of them is right here. It's a restriction point called which is controlled by the RB protein, retinoblastoma protein, which is encoded by this cellular retinoblastoma gene. This controls entry into S. this protein decides whether the cell is going to go into S. And the signals that lead to that we'll get to in a moment, and we'll discuss even more next time. So RB controls entry into S, and it was discovered because young children have retinoblastomas, tumors of the retina. Early in life, they develop these. And these kids have a loss of the gene encoding RB. And so the loss of the gene is associated with tumor formation. Therefore, we have called this a tumor suppressor gene. So its presence is needed to avoid these tumors. And the reason is because it keeps cells from dividing forever. And that's what leads to cancers. When cells start to divide forever, uncontrolled, then they become cancerous by mechanisms which we'll talk about in a couple of lectures. All right, so what does this have to do with viruses? Well, these immediate early proteins, or these early viral proteins, abrogate RB. And we've talked about this before in the context of adenovirus E1A. So here is these pink guys here are these early proteins of viruses, SV40 large T. The papillomavirus has an equivalent, and then there's the E1A protein of adenovirus. And these proteins bind and sequester RB. Now, I told you last time that the adenovirus early transcription requires E2F proteins. These are transcription regulators, normally bound up by RB, and so not available for viral transcription. When RB is bound to E2F and E2F sits on a DNA near a promoter, the RB recruits histone deacetylases, and that tightens up the DNA so it can't be transcribed. So RB is shutting off E2F-controlled promoters. And those promoters, by the way, control genes that are needed for cell division and DNA synthesis. So the cell control is mediated by RB sitting on these promoters, and that's how it works as a checkpoint. So again, these are genes encoding replication proteins of the cell that allow the cell to synthesize DNA and to divide, go through mitosis. Viral promoters, like for adenovirus early promoter, its replication proteins, which are encoded in the early region, are also controlled by E2F responsive promoters. And so that's how the checkpoint in the cell cycle works. RB sits on these promoters until the conditions are right that cells can divide. And those conditions include having adequate nutrients in the cells and so forth. Now, these viruses need to get the cells to divide. So they encode these early proteins that sequester RB. They pull it off E2F, and now these promoters are active. The cells begin to make proteins that are needed for cell division and DNA synthesis, and there are many of those. But that's how the viruses, when they get into a cell, can ensure that the cell is dividing. They have their early proteins bind RB. So these proteins have a lot of functions. Large T already, you see, it's involved in viral DNA synthesis, and it's also abrogating RB so that there are available enzymes for viral DNA replication. So to summarize this, the viral early proteins abrogate RB, so there's no more checkpoint on the cell cycle. Now the restriction point is gone. The cells can divide on and on and on and on. But of course, eventually they'll die because the virus reproduction will kill them. So they won't divide forever. And so that's how the viruses get the cells to enter mitosis if they're not already and produce the proteins that they require for DNA synthesis. It's all at the level of RB. Here you see the cell actually, if it's not virus infected, there are obviously some conditions where the cell has to divide and remove the RB checkpoint, and that's done by phosphorylation by cell cycle specific kinases. Right, so there are some conditions where you have to divide, and that's how we do this in the absence of a viral early protein. Someone's asking, what does DP do? It's simply another part of this transcription protein, which is binding to the promoter. The exact function I'm not sure of, but it's always present in the E2F complex. but RB binds E2F, not DP. And DP alone, it's removing RB from this equation that's important because E2F DP is the active complex. That is our discussion of DNA synthesis. We're actually going to come back to it because this idea that viral proteins can kick cells into dividing has consequences for cell transformation and tumor formation. Next time, we're going to get into a discussion of reverse transcription and integration, and that's the process by which retroviruses and other viruses make DNA copies of their RNAs.